Welcome to Cell by London Training Academy. My name is Dr. Moustafa and I will be teaching the fundamentals of flow cytometry. First, this is the first lecture of this course, which is I'll be talking about the principles of flow cytometry. First of all, we need to learn what does the word flow cytometry mean. Flow cytometry is composed of three parts. Flow, which refers to a liquid or gas or even electricity that moves steadily and continuously in a current or stream. And the word cyto means cell, referred to cell, and this is actually a prefix. And metry is a suffix, comes from the Greek word metria or metrias, which means measurer. So the flow cytometry technique is used to detect and measure physical and chemical properties of a population of cells or particles. A sample of cells or particles will suspended in a fluid and is injected into the flow cytometer instrument. Through a narrow tube, the sample is focused to ideally flow one cell at a time through a laser beam, and the light scattered is characteristic of the cells and their components. When the cells are labeled with fluorescent markers, fluorescence light is first absorbed and then emitted in a band of wavelengths. Tens of thousands of cells can be quickly examined, and the data gathered are processed by a computer. So why actually do we need flow cytometry? A flow cytometry analyzer provides quantifiable data from a sample in addition to flow cytometry cell sorters, which physically separate and purifies the certain cell populations of interest based on their optical properties. There are wide applications of flow cytometry. In general, it is routinely used in basic research, clinical practice, and clinical trials, such as cell counting and sorting, determining cell characteristics and functions, detecting microorganisms, biomarker detection, diagnosis of health disorders or diseases such as blood cancers, and even in protein engineering detection. So how does this machine works? So we need to understand how the machine works, how the flow cytometer works, in order to be able to adjust the settings, able to interpret the histograms and all the information comes out from this machine, and also to be able to get a true results that reflects true information. So every flow cytometer is composed of three components or three systems. The fluidics system, which is in yellow, which is the excitation optics. The fluidics system is the blue ones, which is a sample, the flow chamber, and the sheath fluid. The optics system, which is in the yellow color, the excitation optics and collection optics, and the software, which is the electronic system that take all these data and present it and analyze it. So fluidics system. So for flow cytometer, this is the fluidics system, and flow cytometer instrument measures cells as a single file. When a sample enters a flow cytometer, the particles are randomly distributed in a 3D space through a large space of the sample line. The sample must therefore be ordered. It needs to be ordered into a stream of single particles that can be interrogated individually by the instrument's detection system. This process is managed by the fluidics system. The fluidics system consists of a central core through which the sample fluid is injected, enclosed by an outer sheath fluid. So these two parts with the yellow arrows are the outer sheath fluid, and actually it helps the cell suspension to be ordered in a single cell line. Well, the pathway that fluids follow, it's the flow buffer sample, okay, and then the samples, then the cell flow nozzle, and then collection tube to the waste. So it passes from the sample flow to the nozzle, where the cells become ordered one cell at a time to be measured, and then there is a waste product outside here. So due to narrowing of the sheath in a nozzle or cuvette, the fluid velocity is increased. So this is the sheath fluid, and this is called the cuvette, and the sheath, when it runs, and the fluid velocity increases, and the cells in the sample is introduced into the center and is focused by the Bernoulli effect. This allows the creation of a stream of particles in a single file and is called hydrodynamic focusing. Under optimal conditions, laminar flow, there is no mixing of the central fluid stream and the sheath. Okay, without hydrodynamic focusing, nozzle of the instrument would not create a focus stream of cells, and analysis of single cells would not be possible. With hydrodynamic focusing, the cells flow in a single file through the illumination source called the interrogation point. So the cells here, after it becomes a single file, every cell get in contact with the light source, which is the laser beam, and it's called interrogation point, allowing single cell analysis to get into effect. So this is how it normally works. So here comes the optics part. After hydrodynamic focusing, each particle passes through one or more laser beams, or focused laser beam, of course. Light scattering or fluorescence emission from autofluorescence, or if the particle is labeled with a fluorophore, it provides information about the particle's properties. Lasers produce a single wavelength of light, or you can say a laser line, at specific frequency. They are available at different wavelengths ranging from ultraviolet to far red, and have variable range of power levels as well. So the interaction of light or laser beam with the cells or particles resulted that the light is scattered in different directions. One of the directions is called the forward scattered light, which is typically to 20-degree offset from the laser beam's axis, and is collected by a detector called photomultiplier, or PMT, known as the forward scattered channel. So the angle, however, vary depending on your instrument, leading to variation of forward scattered channel signals between different machines. That's why when you measure when you're doing an experiment with 3 repeats, you must use the same machine every time you do the repeat in order to avoid any variation of the results. So why do we need forward scatter measurements? Actually, forward scatter measurements can give an estimation of the particle size, with the large particles refracting more light than the smaller ones. So when the particles are smaller than the wavelengths of the illumination source, let's say 200 nanometer exosome, and it's passing through a 488 nanometer laser, it doesn't necessarily scatter light in a forward direction. So this is how we call it a forward scattered light. It's the light scattered at the angle of 20 degrees from the laser beam's axis, and it's, in general, to sum up, it gives the estimation of the particle size. Another type of scattered light is the side scatter, but before that I'll just show you how it works when the cells are measured. So when the cells are measured, the computer can detect the forward scattered measurements, giving an estimation of a particle size based on the amount of light reflected, and that's how it is represented in the form of histograms. So if you show it again, so if it's a big particle, it will give you a big graph, and every single particle is dotted and plotted on this histogram on the computer, and you can read the forward scatter measurements. Another type of measurement is the size scattered channel, and this actually it's a light measured at 90 degrees. So when the light scatters, when it comes with when it comes in the interrogation point with the laser line, then it refracted at 90 degrees, and it refracted at 90 degrees, and this is, in this case, it's called the side scatter channel. It actually provides information about the relative complexity, for example, granularity and the internal structure of the cells or particles. So flow cytometry converts the amount of side scattered light into a voltage pulse, which is then plotted by the computer as side scattered. So both forward scattered and side scattered channels are unique for every particle, and a combination of the two may be used to roughly differentiate cell types in a heterogeneous population such as blood. So they give you the the sum of the forward scattered channels give you the identification of the size or estimation of the size of the cells, and the side scattered channels give you estimation of the granularity and the texture of the cell. We have another thing here, which is when you see it on a histogram, so you will be able, because the computer analyzes the side scattered channels and forward scattered channels, depending on the sample type and the quality of the sample preparation. It's actually fluorescent labels are generally required to obtain more detailed information. So analyzing side scattered channels and forward scattered channels helps the researchers understand different cell populations through the size, shape, and complexity. So normally fluorescence measurements are taken at different wavelengths and can provide quantitative and qualitative data about fluorophore labeled cell surface receptors or intracellular molecules such as DNA and cytokines. Most flow cytometers use separate channels and detectors to detect emitted light, the number of which vary according to the instrument and the manufacturer. That's why, again, when you use when you're doing repeats, you have to make sure that the 3 repeats are done on the same instrument. The specificity of detection is controlled by optical filters, which block certain wavelengths whilst passing others. There are 3 major filter types. Long pass filters allow light through above a cutoff wavelengths. Short pass filters permit light below a certain wavelength, and band pass filters transmit light within a specific narrow range of wavelengths, termed as bandwidth. So these filters can block light by phased reflection, allowing certain light to pass through and interfering with other wavelengths. So how can a signal be converted into a measurement? When a particle passes through the interrogation point, it generates a signal which is detected as a pulse, known as event, in every detector. These pulses reflect the passage of the particle through the laser beam or beams, and the signal generated at each point in the cell's path. These pulses can be mapped by plotting signal as a function of time. As the particle enters the laser beam spot, it will generate scattered light and fluorescent signals, which will ultimately manifest in a current of electrons from the anode of the detector, known as the PMT, the photo multiplier detectors tubes, which are the detector in general, and the photons that hit the photocathode, and thus is also proportional to the intensity of the scatter or fluorescence signals generated by the particle. the output of the PMT detector will begin to rise, reaching peak output when the particle is located in the center of the laser beam. At this point, the particle is fully illuminated, which means the laser beam's photons are at highest density and will produce a maximum amount of optical signal. As the particle flows out of the laser beam, the current output of the PMT detector will drop back to the basal line. So here you see on the graph, this is the height or the maximum amount of current output by the detector, and this is the area of interrogation the integral of the pulse, which shows the intensity of the pulse. And as for the signal intensity, can be measured either by the area or the height. So this is the area or the height where you can know the intensity of the signal. And the width here is the time interval, which is the time, which that can be measured by either a high. This is the time which is during which the pulse occurs. The width parameter measures the time that the cells spend in the laser beam. So the time the cells, every single cell, spend on the laser beam. This shows the time. Okay, so there is one thing that you need to learn about, is that when the cells come in contact, every single cell, when it comes in contact of the laser beam, there are some cells which are dead cells, or you can say debris. So not all the signals that are generated correspond to a particle of interest. Actually, the PMT detectors are extremely sensitive and detect signal from a variety of sources that are irrelevant to the experimental data, including stray of light, dust, cell debris, and things like that. So in this case, these datasets would mask out relevant data points and overload the electronic ability to process relevant signals. And in order to avoid processing these unwanted signals, measurements are taken based on the signal intensity, or what we call threshold. So this is done by designating a parameter as the trigger for recording events, usually for forward scatter, and setting a level in that parameter as the threshold, because normally these cell debris or the stray of light or dust or whatever, these kind of things are small in size. So there is a minimum level that you have to get in order for your results to reflect a true result, and this is called the threshold. Okay, so any pulse below the threshold level, this is ignored in all detectors. Any pulse that is above the threshold levels is fully processed by the electronics. So this is how you can adjust the settings of your experiment or your flow cytometer in order to reflect the true results, rather than having signals of everything detected through the laser beam. So data is collected actually in a linear scale, but usually displayed as log scale for fluorescence studies, because it expands the weak signals and compresses strong signals, resulting in a distribution that is easy to display on a histogram. Linear display is required when very small differences in fluorescence signals must be assessed. For example, in DNA analysis, where there may be two-fold increase in fluorescence. The measurement from each detector is referred to as a parameter. So each parameter can be displayed in height, area, width, and width values, and on the histograms and dot plots in the flow cytometry software. These are used to measure fluorescence intensity, compare populations, and designate sorting decisions. So in the next lecture, I will talk about fluorescent markers, fluorescent proteins, discuss common fluorophores used in flow cytometry.